So everything that we're going to do for the next few weeks will all be in chapter 17 of your book, and we will more or less be going in the order that your textbook goes in, with a few small exceptions. The first lesson, which I called P's, Patterns, and Probabilities, all comes from section 17.1, so the first section. Now I'll talk about the P's and the patterns in a minute, but I do want to focus on the word probability right now, even though we're going to come back to it again and again. Everything that we're going to do in terms of predicting what will happen in offspring, which are the descendants or the babies that organisms produce, is always a probability. We are not predicting certain events. We are just predicting what is the most probable thing that would happen if these are the two parents. And so I really want to highlight that these are probabilities. This is what is probably going to happen, but there are cases where something that is improbable happens. So for example, it is improbable to find a family full of people with brown eyes and brown hair, and then to all of a sudden have a blonde-haired, blue-eyed baby. But it is not impossible. It's simply improbable. And so I want to really focus on the difference between impossible and improbable, because we're talking about probabilities here. What is likely going to happen? So the first thing that we will talk about really is how did we get to where we are? I think that it would be unlikely that you would have to identify all of these scientists and their theory. I'm trying to show you a progression here so that you understand we didn't always know the things that we know now. Some of you might remember hearing about Aristotle, maybe in Science 10 when you're talking about cells, or maybe in Bio30 when you were talking about evolution. But Aristotle was of the opinion that organisms were just the way that they are, and that's how they were made, and there was no change over time. When they were born, that was the way they were supposed to be. Van Leeuwenhoek, who you also might recognize, Microscopes, Science 10, he came several hundred, almost several thousand years later, and he built a microscope and was looking at things. And his idea was that the way that you got characteristics was because a sperm cell had a homunculus in it. Does anyone know what a homunculus is? It's just a fun word to say. A homunculus is a perfectly formed miniature human. If you watch The Big Bang, they call Leonard a homunculus because he's short. But essentially, it's like a miniature version of a person, but with the exact same proportions as a regular person. So his idea was that the sperm had this, and that is where characteristics came from. Another guy, de Graaf, came along later and said, no, I actually think it's the egg that contains the entire person, not the sperm. So we had these competing ideas. The other thing that people have known for centuries, probably even longer than we've had ideas about cells and the origin of cells, is that selective breeding is possible. I put some pictures there of dogs because I think probably most of you are familiar with the idea that we can get dogs of different appearances by choosing who their parents are. So for example, at the end, a Labradoodle, I think is the name of that thing. Mine is a miniature dachshund. That's this little sweet girl right here. Miniatures often get made by selecting two parents who are also small from a larger line of that race of dog. You have the Grand Pyrenees, which was selected for its size and sort of durability. And then there's a picture of a wolf. You might remember or have heard that it seems as though dogs and wolves and coyotes and all of those things probably have a common ancestor. And so we know that there are different ways of getting different characteristics based on who reproduces with who. These are just some ideas that people have had over the years. It wasn't until a guy named Mendel, Gregor Mendel, came along and spent a huge chunk of his life growing plants and studying them, that we came up with some more specific ideas about how to get characteristics specifically. So Mendel was a monk. He lived in a monastery. He had this giant garden full of pea plants. So when I called this lesson Peas, Patterns, and Probabilities, it's because a lot of our initial information about genes and genetics and how things were passed on comes from Mendel's work. This information is on page 588 of your textbook. You do not have to memorize it. I'm just going to use it as my example because it's what Mendel studied. Mendel was looking at peas, so little green peas, you eat them. And he grew a wide variety of different plants that had different traits. If you look, he studied a bunch of different characteristics of pea plants. Flower color, flower position, seed color, seed shape, pod color, and stem length. All of these characteristics were visible, and what he did was took plants with different traits and crossed them. In other words, made them reproduce with each other, and then looked at what happened. There are some terms that we're going to use, so we need to have some definitions of them. Mendel always used true breeding plants. True breeding plants always have 2 copies of the same allele. Now the word allele we used a while ago, and we're going to start using it again. An allele is a version of a gene. So for example, you have a gene for eye color. You actually have a few, but I'm just going to assume there's one. And there's the blue version or the dark colored eye version. Those are the alleles. True breeding plants have 2 copies of the same allele. The way you ensure that that's true is by auto-fertilizing the same plant over and over again to make sure that there's no possibility of having a different allele. Mendel did a whole bunch, thousands, of monohybrid crosses. Mono means one. And so he would pick 2 pea plants that differed, so you see the word hybrid here, on one trait. So he would pick 2 pea plants where everything was the same except one of them had purple flowers and one of them had white flowers. And it was really important that he was picking plants where only one thing was different at a time so that he could control things. So he did a really good job of controlling things in his experiments and making sure there was only one thing that was different. So in a monohybrid cross, we take 2 plants, in this case, with one trait that differs. Other than that, everything else was the same. That allowed him to study just that one particular trait. What we're going to see is that for now, we're going to stick with monohybrid crosses, where we're just talking about one trait. But as we progress, we're going to add other characteristics as well. When Mendel was representing his crosses, and we will use this notation as well, we always referred to things by which generation they are. The parental generation, which is represented with the P, you can see up here, this is the first cross. So in other words, it's the beginning. Yes, those plants came from other plants, but when we're considering which characteristics they have, they are the ones that we are starting with. So the parental generation will always come first, and it's usually in a question what we will tell you about. The next generation, you can see down here, is called the filial generation, and perhaps that word seems unusual to you. If you have ever taken French, you might know that to say son or daughter, you say fils or fille. I think when you talk about horses, there's isn't one of them called a filly or something like that? But it's a word that means that we're talking about the offspring, or we might use the word descendants. Or if it's humans, we might use the word babies. If we're talking about animals, you'll actually see that in a lot of questions, we use the actual word for that baby animal, like a baby cat is a kitten, a baby dog is a puppy. And so you'll recognize a lot of those words, I think probably because they're familiar to you. If we ever encounter a word that might not be familiar, like an ocelot, maybe you don't know what that is a baby owl, I would tell you that's what it means, okay? Yeah. Isn't an ocelot a baby? Maybe I'm mixing them up. Okay, it's a baby something. Maybe I'm mixing them up then. It's a word that starts with an O, though. Maybe the French word sounds different. See, you guys, here's my problem. I switched between two languages. I'm so mixed up. You gonna look it up? Look it up. What's a baby owl? What's a baby owl then? Oh, it's an owlet. Oh, like on PJ Masks, of course. How did I not know that? Anyways, that's not the point of this. The point of this is you don't have to know those words. If we use a weird word like that, we'll say so in the question what it is. So Mendel is studying these pea plants, and he discovered that when he took two true breeding plants that had two copies of the same allele, and he performed a monohybrid cross, so you can see his cross up here, and he looked at the filial generation that came from that parental generation, something happened that maybe was unexpected. If you take a look at all of the characteristics that Mendel studied, look at the F1. F1 means it's the first filial generation. You'll see that in some questions, we'll talk about an F2. That means we're going to make another generation afterwards. But all of the Fs represent offspring. Notice that only one of the traits shows up. Hey? If I mix a purple and a white flower, all the offspring are purple. If I mix an axial and a terminal flower position plant, all of the flower positions are axial. If I mix a yellow and a green seed color, all of the seeds are yellow. And so he noticed that it seemed like only one of the two traits was showing up. So we could start to talk about dominant and recessive genes. Now, I picked one characteristic that Mendel studied. One of the characteristics that he studied was whether or not the seed was round or whether it was wrinkled. So that was one of the things from the table on the page before. And so Mendel noticed if in the parental generation I had true breeding plants, and I can see their true breeding, they have two copies of the same gene. Now, up until here, you might not know that these letters represent something, and we're going to talk about the letters in just a second. But I know that that round seed has two copies of the round allele, and the wrinkled one has two copies of the wrinkled allele. I can see one's two capitals, one's two lowercase letters. And he noticed that in the F1 generation, they were all round. There were no wrinkled seeds to be found. No matter how many plants he produced, this is showing one plant, but he would have done this and produced hundreds of plants, and he would have noticed none of them had wrinkled seeds. They all had round seeds. And so his conclusion was that, well, there must be something about being round or being wrinkled that's causing this difference. So we need to define dominant and recessive genes. Something that is dominant shows up in the F1 generation. If one of the parents had it, then the offspring have it as well. According to Mendel, something that was recessive does not show up in the F1 generation. Now, these are Mendel's definitions because he always did true breeding crosses where the two parents always had two copies of the same allele. So according to Mendel, if the trait showed up in the F1 generation, that meant that it's dominant. And if the trait didn't show up in the F1 generation, that means that it's recessive. We're going to start by talking only about complete dominance. In complete dominance, one allele completely masks the other. There will be examples later where this isn't true, but for now, this is what we're going to discuss. So in Mendel's pea plants, I don't know what just happened there, when he took two true breeding plants, one with round seeds, one with wrinkled seeds, he saw that all of the offspring had round seeds, and perfectly round seeds, not a little bit wrinkled, but mostly round. And so when I'm saying one allele completely masks the other, I don't see wrinkles at all. There's no evidence at all in the F1 generation that there's any sort of wrinkly allele present. So we're going to talk about things being dominant and things being recessive. And this is a method that was used and is still used to figure out which thing is dominant and which thing is recessive. In a lot of questions, we're just going to tell you this thing is dominant, this thing is recessive. This is how we know. Now, does anyone have anything they want to ask here? So, we are going to talk about the first of Mendel's laws. He has two. And his first law is the law of segregation. And once I show you what this law is about, you're going to say, oh, well, this is exactly what happens during cell division. So this is really not new. It's just a different way of looking at things. In the law of segregation, we're saying that each gamete only gets one of the two alleles a parent has. Now this makes sense. When we undergo cell division, and specifically we're going to think about meiosis because we're talking about gametes here, gametes are haploid. They get half of the parent's chromosomes. Since genes are little sections of chromosomes, if I'm splitting up the chromosomes, I'm splitting up the genes as well. So each gamete only gets one of the two alleles a parent has. So in my little example here, I took the F1 plant from the previous example. That plant had one round and one wrinkled allele. Those were the two alleles that it had. If I want to represent which gametes could this parent potentially make, a typical way of doing this would be to use a little square called a Punnett square. And some of you might have seen these before. I know in grade 9 there's like a little section about heritable characteristics. But if this is new to you, don't worry, we'll talk about it. When I think about possible gametes, and we'll include the word possible here, and I take those plants from the F1 generation, and I take one male and one female plant. Since they're plants, it's okay for a brother plant and a sister plant to have baby plants together. Yeah. It happens all the time in nature because plants don't know that they're brothers and sisters. And I think about what gametes they could possibly make. The female plant could make a gamete that has the round allele, or she could make a gamete that has the wrinkled allele. And the male plant could do the same. You might say to yourself, hey, I'm sorry, I thought that in spermatogenesis you made 4 sperm cells, and you do. These are just possible combinations. So I could write 4 copies of this, but all I'm saying is what are the chances? What's the probability? If I consider just this characteristic that's controlled by this gene, there are 2 choices for the sperm. Either the sperm has the round allele or it has the wrinkled allele. Doesn't matter how many sperm cells I make, those are the 2 choices that I have. Same thing for the female. I know that in oogenesis, we're only making 1 egg at a time, but in that 1 egg she's going to make, there are 2 possibilities for which allele is going to show up, either the round or the wrinkled. Now, so far, does anyone have anything that they would like to ask? Okay, okay folks, we need to talk about this awesome thing called a Punnett square because it is a really useful method to use when representing a cross between 2 organisms. When we consider a Punnett square, there are some words that we're going to need to be able to use and recognize to set it up. Before we can even make a Punnett square, we have to know the genotype, which potentially will tell us the phenotype of an organism. The genotype is which alleles do they have? So if we consider Mendel's pea plants, and there's that table in your textbook, you do not have to memorize it, but I'm going to use Mendel as my example for now. If I consider 2 different possible plants that Mendel might have used, one of the characteristics he studied was flower color. There were either purple or white flowers. So parent 1, in my little example, is going to have 1 capital P and 1 little p. Parent 2 is going to have 2 little p's. Now, why did I choose letter P? Because the dominant color was purple in this example. So one of the things that you'll notice, and this will be represented kind of everywhere, when you have a lowercase letter, it means the recessive allele. And when you have an uppercase, it means the dominant allele. That's the important part. Which letter of the alphabet you use is completely irrelevant. Sometimes the question will tell you which letter to use, like they'll say capital P, little p. You can pick whatever letter you want. I like to use M's a lot because it's my name. Sometimes I like to use Q because I feel like it's a neglected letter of the alphabet, same thing with J. But the actual letter has no relevance whatsoever. It's just a way to represent things. So in my little example, I have 1 parent who has 2 different alleles, 1 dominant, 1 recessive, and 1 parent who has 2 of the same allele, 2 recessives. The phenotype asks you, what is their appearance? With that little a that I added because I was going to spell it wrong. So what do they look like? Which trait do they actually have? Because genotype just tells you what alleles they have, but it doesn't necessarily tell me what they look like. I have to add that in. So parent 1 would have purple flowers. How do I know this? Well, dominant means it shows up. So since this plant, has a copy of that dominant purple allele, that's the color the flowers are going to be. Mendel discovered there was complete dominance. The flower wasn't mostly purple and a little bit white, it was all purple. Parent 2 will have a white phenotype. Why? Parent 2 only has the recessive allele. There's no copy of that dominant allele that would give purple flowers. So this particular plant is going to have white flowers. Questions vary based on whether they ask you for the genotype or the phenotype. Sometimes we're going to ask you, what alleles does someone have? Sometimes we'll ask you, what do they look like? There are two other words that go along with this discussion, and they are the words homozygous and heterozygous. You might recognize the beginnings of the words. Where essentially the prefix homo means the same and hetero means different. If we say that an organism is homozygous, that means that in the zygote that made them, they were two copies of the same allele. So I would say in my example that parent 2 is homozygous. They're homozygous for the recessive allele, but they have two of the same allele. Heterozygous is where the organism has two different alleles. And in my example, parent 1 is heterozygous. They have one copy of the dominant allele and one copy of the recessive allele. So now that we know all of these words, we would be ready to answer a question about these two parent plants. So perhaps my question is, if I have a heterozygous purple plant and a white plant, what are the possible offspring? That's more or less what the question would sound like. So you had to know what heterozygous meant. You had to know how the phenotype related to the genotype. So you had to know a lot of information. If that's the question, these are my two parents, and I'm going to cross them. Here's what I'd start by doing. On my Punnett square, I always put gametes along the top and along the left side. If you really want to, put them on the bottom and on the right side. This is just a method. So I go from the top and the left because that's how you read words in English. But we're going to put the gametes along the side. Remember that gametes, possible gametes, have half of what the parents had. There's segregation that's happening. So I'm going to put parent 1 up here and parent 2 down here. Parent 1 could make two different gametes. Their gamete could have the dominant purple allele or the recessive white allele, because that parent has both of them. There's no way of knowing which one we're going to get. Parent 2, which I'll put down here, only has the recessive allele. So they could make a gamete with the recessive allele, or they could make a gamete with the recessive allele. That's the only option for this particular trait. So notice, what I put on the outside of the square is always half of what the parents have. Then I start filling them in. It is common practice to put the capital letter first. There's no rule, but it's commonly what I do, and it's commonly what you'll see in other questions. When I filled in that top box, I looked what was above it and what was beside it, and I put those two letters together in a box. Each box represents a possible offspring. Not a for sure, but a possible offspring. I could go across and fill in the next box. That one would have a little p and another little p, because if I look to the left and above, those are the letters that I get. Now, you'll notice that the bottom is shaded, and it wasn't on purpose at first, because this is just how PowerPoint makes tables, but then I was like, oh, hey, it's actually really useful. I could stop here. Why? I'm going to get the exact same answers in the bottom, because I have the exact same allele combination for parent 2. So if I consider just these top two boxes, what would I say are the possibilities for the offspring? So remember, each of these is a possible offspring. Not certain, just possible. Well, I would say, well, there are two of them. 1 out of 2 or 50% or 0.5, which all mean the exact same thing, would be purple. So I could say 50% of the offspring will have purple phenotype, or I could say 50% of the offspring will have a heterozygous genotype. I could say either thing and I would be giving you the same information. I could also say the opposite. I could say 50% of the offspring, or 0.5, or 1/2, will be homozygous for the recessive allele as their genotype, or white as their phenotype. Right? I could say either of those things. If I filled in the bottom, would my answer change? No. I would say 2 out of 4, or 50%, or 0.5, would be purple. 2 out of 4, or 0%, or 0.5, or 50% would be purple. So what I'm saying is this. If you feel comfortable and you recognize, oh, I've got 2 of the same allele, I'm not going to make another column or another row in my Punnett square, don't. Some people don't feel comfortable in the beginning, and they like to always represent what are the 2 gametes parent 1 can make, and what are the 2 gametes parent 2 can make, and then worry about it afterwards. And if you are feeling at all uncertain, do that in the beginning, and don't start omitting things from your Punnett square until you feel like, yes, I got this and I know what I'm doing. Now, in terms of how will I ask you to represent answers, I will tell you if I want a fraction. Oh, of course this is what I want. Sure, Smartboard, go ahead. There you go. I'll tell you what I'm looking for. If it doesn't matter to me how you represent your answer, then I won't say, please represent it as a percentage, and I would accept 1/2, 50%, or 0.5, because they all mean the same thing. The only time it really makes a difference is if it's a numeric response question, especially on the diploma. If it's something we're doing in class, I can usually be quite flexible and say, oh yeah, 0.5 is 50%, I gotcha. But on the diploma, they will not do that, and they will tell you, represent your answer as a percentage, or represent your answer as a decimal number, and you just have to be careful which one they're asking for. It will always say which one you have to use.